Hi, I am Dr. Visula Surya, Senior Lecturer in Immunology at Institution of Biochemistry, Molecular Biology and Biotechnology, University of Colombo. In my series of Future of Life program, today I would like to bring to you another very interesting scientific finding. Actually, the scientists who involved in this technique and find this technique has received Nobel Prize in 1984. This technique is called Hybridoma. This is the birth of monoclonal antibody. When I tell these scientific names, you may little confuse. But don't worry, I am going to explain in very simple language what is hybridoma, what is monoclonal antibodies, and how this help to mankind. Before I give explanation of hybridoma and monoclonal antibody, we will see our immune system. As I explained in my previous sessions, our immune system mainly consists of innate immune system, adaptive immune system, and complement pathways. Innate immune system is very quick, but it is not very specific. When you consider adaptive immune system, it is very specific and lasts long. Now, when you think of this adaptive immune system, it mainly consists of cellular immunity and humoral immunity. What is the meaning of cellular immunity? Cellular immunity mainly consists of T lymphocytes. These are special white blood cells. There are few main types of T lymphocytes. We call them cytotoxic T cells. We know them as CD8+. Then we have T helper cells. We nomenclate them as CD4+. Then you have the T regulatory cells. For example, we call FOXP3. And you have the T memory cells. So when you consider the humoral immunity, it mainly consists of B lymphocytes. This B lymphocyte, when it activates, it can proliferate into plasma cells and memory cells. These plasma cells are very important. Their prime work is to produce antibodies. Millions of different types of antibodies they can produce. Basically, whatever pathogens, example, viruses, fungi, parasites, basically all of them can covered by these antibodies. It has a vast variety. And some part will remain as memory cells. It is very important. The similar pathogen invade the body again, the memory cell will quickly identify and start attacking them. Now, when this natural system works, these plasma cells or B cells will produce lot of antibodies. It is actually mixture of antibodies. Those mixture of antibodies we call polyclonal antibodies. Different B cell clones will produce different antibodies. That is the meaning of polyclonal antibodies. But they are not so much specific as monoclonal antibodies. Monoclonal antibodies mean selected single clone of B cells will produce a specific antibody which targets a specific antigen. It is very target oriented. Now, how to do this? So, these Nobel Prize winning scientists, they have take rats and put a special or wanted antigen to their bodies and kept them, induce immunogenesis. And after weeks, they take off the spleen where the B cells are colonized and extract the B cells. Once you extract the B cells, there's a problem of these B cells. That is, these B cells are not long lasting, even though they produce antibodies. So what they do, they take myeloma cells. Myeloma is a cancer cell, which is last immortally. That means it can divide many times without dying. So what they do, they use a special chemical that is called PEG and bind this myeloma cell and B cells. This binding we call hybridoma. Now I think you will understand what is the meaning of hybridoma. Then what they do, there's a special media called HAT. They put these cells into HAT media. If this B cells and myeloma cell bind, if they produce a hybridoma, it will last. The remaining B cells, unattached B cells and myeloma cells will die. Then what you do, from this they will select specific antibody what we need and they colonize and obtain the antibodies. These antibodies are coming from a single hybridoma beta B cells. So these are called monoclonal antibodies. So what is the use of this monoclonal antibodies? It has three main uses. Mainly, if you think of mankind, it serves as a treating method, treating modality. Mainly, autoimmune diseases like SLE, rheumatoid arthritis. Then myasthenia gravis. It is a nerve system disease associated with nerve system. Then you have diseases like COVID. Then you have apart from that, it can treat cancer cells. That is another most important thing. Second thing is, it will help you for diagnostic modalities like HIV, like cancer markers. It will help screen human having these diseases. Last one is, it helps for research. Mainly the immunoblotting, Western blotting techniques, ELISA techniques, these monoclonal antibodies will be used. So you can see this marvelous discovery of hybridoma is the birth of monoclonal antibodies. So it has a large impact on human being, present as well as future. I hope this brief introduction on hybridoma and monoclonal antibodies and its benefit to humankind will enlighten your knowledge. So thank you very much. Stay safe and stay healthy.